Thank you, Patty. Next, I'd like to welcome Dr. Angie Pinto, who was mentored clinically by David and was also one of his PhD students. Is continuing her studies now. Thank you, Angie.
Thank you to the organisers for asking me to speak, and it's truly an honour to present today at this symposium as one of David's current PhD students. The end of HIV, this is what we've all been working towards. I'd like to start by reflecting on this editorial that David and I wrote in the MJA in 2014 at the time of the International AIDS Conference. While we talked about treatment as prevention and vaccine development, David made a point to include the following statement to ensure treatment and prevention access for all, stating that Australia should continue its commitment to support countries in our region to ensure access to treatment, reduce detectable viral load, and ultimately stop HIV transmission. So what I'd like to talk about today is another aspect of prevention, combining molecular genetic techniques and epidemiology to understand and ultimately stop HIV transmission. So many would be familiar with molecular epidemiology in terms of understanding the origins of viruses and where they come from. This is a phylogenetic tree of HIV and shows its genetically related viruses, which allowed us to understand that its origins from viruses found in primates in Central and West Africa. Today, we can use the genetic sequence of a virus in conjunction with clinical and epidemiological data to gain a great understanding of epidemics from things like subtype diversity, drug resistance mutation, and transmission dynamics. So that is understanding how the virus is transmitted within populations and where the virus is going to. So what I'd like to do today is illustrate some of the insights molecular epidemiology can provide as we move towards the virtual elimination of HIV. You can see here there's been a growth in this field over the years, and many would agree that David was ahead of his time, as he was thinking about this area for many years ago, although the landscape in Australia wasn't quite ready to embrace this type of research. You can see the solid lines show the growth in the number of articles published around HIV transmission clusters, which corresponds to the availability of HIV sequence data in the grey bars. So transmission clusters can be defined in a number of ways, but in general relates to how similar the genetic sequence of a virus is and is based on the number of mutations that differ from the parent virus. I'd like to share with you and set the scene for this research some of the articles that David sent to me over the years. And some people have reflected on David's uncanny ability to be able to extract a pertinent journal article from the depths of the available literature and send it to very carefully selected recipients. And this is an example of some of these. These articles highlight how different jurisdictions have used molecular epidata to understand their local epidemics. And they've looked at things like transmission dynamics and networks to tell us how the virus is transmitted within populations. Others show the use of phylogenetics to guide prevention efforts and break transmission cycles. So some in the audience may be familiar with this work from British Columbia and Canada, which was a population-based phylogenetic analysis which included half the population of HIV infected in BC. So firstly, they defined transmission clusters, which you can see here with cluster size indicated by the size of the circles. And you can see that these are largely separated by risk group, with men who have sex with men in red and people who inject drugs in blue. On the right-hand side, they showed cluster growth over time, and what you can see, stratified by risk group, is some clusters undergoing particularly rapid expansion. And these types of clusters are those that would be potential targets for public health interventions. Next, they looked at some of the factors associated with cluster membership, and David remarked, "Great data from Vancouver on cluster analysis. Fascinating to see that clusters are related to viral load. Guess it reflects recency of initial infection, with a higher mean viral load and acute infection being more significantly associated with cluster membership. And this sort of information provides reinforcement for early diagnosis and early treatment." This was another article called "Connecting the Dots." And this outlines some of the various strategies to define networks. On the left-hand side, you can see some of the limitations of phylogenetics methods alone to define transmission networks, in that there may be missing links from individuals sampled in orange versus another individual who may not be sampled, for which we don't have the sequence. There's also limitations in terms of inferring directionality, and there's no way to know by phylogenetics alone in which direction the transmission might have occurred. On the right-hand side, we can enhance this data with the use of social or behavioural data, forming a complete network, as well as using things like dates and epidemiological data to understand directionality in conjunction with phylogenetics. However, this also has limitations too, and you can see that many different topologies can be inferred despite using all of these methods. So David remarked, this is a good review in AIDS looking at the advantages and disadvantages of mapping networks through social science, public health and phylogenetics. Any suggestions on using these approaches in our various studies would be welcome. And this type of thinking highlights a truly collaborative approach and multidisciplinary approach fostered by the Kirby Institute that makes its research so relevant and successful. And I'd like to give you an example of one of those approaches today. So this is the New South Wales HIV Prevention Project, which is a collaborative research partnership with funding for the NHMRC, New South Wales Health and University of New South Wales. And it's comprised of 4 main pillars underpinned by the New South Wales HIV strategy. The Molecular Epidemiology Working Group is a subgroup of the treatment pillar and aims to help inform various aspects of this framework, from treatment impact through drug-resistant surveillance to evaluating the impact of testing and prevention strategies, and providing information on some of those key groups at risk of transmission or those that are getting left behind from the current strategies. So here are some of the collaborating partners who have helped with the extensive consultation process involving legal input, community engagement, and appropriate ethical and privacy considerations. So what we've found is we've looked at transmission clusters in New South Wales, and you can see here a de-identified phylogenetic tree of subtype B sequences in New South Wales, showing those that belong to clusters in coloured tips. And we looked at some of the factors associated with these clusters, and we found that they tended to be of younger age, acquired in Australia, early clinical stage, and more recent calendar year of diagnosis, suggesting that the current public health interventions are on track, as well as potentially even the contact tracing measures picking up these clusters at an early stage. This is work done by Francesca Di Giuliano, and she's looking at trying to map the shape of the epidemic over time. On the left-hand side, you can see a phylogenetic tree of subtype B HIV sequences, and the clusters are indicated by different colours. On the right-hand side, you can see the cluster length by calendar year. And what we found is one of the largest clusters, marked by the blue star here, had up to 26 sequences, and one of the longest lasting clusters has been active for over 22 years. Now, this is still a work in progress as part of this partnership grant to 2020. And what really remains to be seen is what will be the impact of PrEP going forward. This analysis is just to 2016. That's one of the key questions we want to look at. So in summary, this was a glimpse into some of the insights molecular epidemiology can provide in ending HIV transmission by understanding the impact of public health interventions on cluster growth. But there's also a couple of other areas I haven't touched on, including subtype diversity, and there's been some good work done around this by the Australian Molecular Epidemiology Network, and also around surveillance and establishing surveillance systems for drug resistance mutations, particularly in the era of PrEP rollout. This is really using population-level data to inform public health interventions and is part of an ongoing collaborative research framework. And I'd like to acknowledge my supervisors, David, of course, and also Kathy and Tony, who've been an incredible support during my candidature. Also, there's many collaborators listed here without whom this research would not have been possible. Now, this wouldn't be a molecular epi talk without a phylogenetic tree. And I'd just like to pay tribute to some of these clinician researchers who've had an impact on me over my training, either as supervisors, mentors, colleagues, or now peers. And what they all have in common is they've all once been research students of David's, with the years of their submitted degrees listed there. And it's really a testament to the fact that David was a great supporter of young clinicians, and really it shows his passion for clinical research. But this is just a handful, and what you see, the last slide is not showing, but it was a bookshelf of three bookshelves worth of photos stitched together of it's coming up here, of completed theses. And thanks to Jeanette, who helped me put that together. For some reason, it's not projecting on the screen. There you go. So that's three bookshelves worth of completed theses, which are proudly displayed in the front of David's office. And what was striking to me was that the last bookshelf was actually half empty. And I think that David would be very pleased to know, even though he won't be here to see it, that I'm sure that this bookshelf will be filled in the years to come. Thank you.